Welcome to solverfy.
Okay, how is shotgun sequencing performed? So the first step in doing shotgun sequencing, you take a DNA molecule, a large DNA molecule of interest, and you break it down into small pieces, into small fragments of DNA. Then each one of these fragments is sequenced using the chain termination method. What is the chain termination method? It is a method where you take the target sequence and you replicate it, but along with regular nucleotides, you add dideoxynucleotide triphosphates, and dideoxynucleotide triphosphates. So deoxyribonucleic acid. So DNA is called deoxyribonucleic acid. It's like RNA, but it's deoxic. It's missing one oxygen. DD nucleotides have dideoxy, so they are missing two oxygens. And the removal of that second oxygen means that when this DDNTP is incorporated in the chain, replication stops. So DDNTPs block replication. So when you add some DDNTPs along with regular nucleotides in a chain, those DDNTPs will randomly get inserted into the chain at different points, and every time they get inserted into the chain, they stop the replication of the chain. And now that you have all of these different chains, you can then determine, okay, so if the chain is one nucleotide long, that means that the last nucleotide that was inserted was a DDNTP, and you know the identity of that nucleotide through some sort of labeling, right? So then if it's two amino acids long, well, you found out the first time, I'm sorry, not amino acids, nucleotides, I apologize. If it's two nucleotides long, well, you found out the first nucleotide by the one nucleotide length fragment, and now if the last residue uh, of a two nucleotide long chain is T, then you know that the second residue is T. So then you do this for all of the fragments. So all of the fragments get sequenced in this way. And then once you have all of the different sequences for fragments, you line them up and find overlapping regions. And then from that overlapping region, you can form the sequence of the entire DNA. Okay, so let's look at our answer choices. Choice A, the DNA is cut into fragments. Yes. Sequences is done using the chain termination method. Yes. Fragments are analyzed to see overlapping sequences. Yes. And then the entire fragment is reformed. So choice A has the correct order of the steps here. Let's see why the other ones are wrong. Choice B, the DNA is cut into fragments, overlapping sequences are analyzed, and then sequencing is done. No, sequencing is done first, and then the sequence overlaps are determined. So those two steps are wrong. Choice C, the DNA is cut into fragments, stained with fluorescent dye. There's no staining with fluorescent dye, so that's wrong. Choice D, the DNA is cut into fragments, sequencing is done using the chain termination method. So far, so good. The DNA is stained with fluorescent dye. Again, DNA is not stained, so choice D is also wrong for the same reason as choice C. So the correct answer is A. You cut the DNA into fragments, you sequence all of those fragments using the chain termination method, you find out what the overlaps are between the fragments, and then from that you can form the entire fragment sequence.